this meeting wouldn't have been possible without a very generous contribution from the Agouron Institute, and I think we need to really thank them for their very generous support. We also have support from Agrisera, WALZ, SpectroLogiX, and I guess that's it. Yeah. So that's the financial support that allowed us all to come here.
Thanks, Bob. So we are all here to advance the search for life, and for this workshop, especially to scrutinize the diversity of photosynthesis as a producer of biosignatures, that is, signals or phenomena that demonstrate evidence of life. So for this workshop, we're defining photosynthesis broadly to mean biological light harvesting for energy. So it doesn't have to be autotrophic necessarily, but it should show evidence, something that could show evidence of life. And the emphasis in this workshop is very much on remote observations by space telescopes of exoplanets. We're not ruling out, say, fossil evidence of photosynthesis or life on Mars, or even currently in the clouds of Venus, or theoretical ideas about methane-based photosynthesis on Titan. And there are some people here who have some research bearing on those, but our emphasis is on exoplanet observations because we have flagship missions coming up that are being designed by the community now, the Habitable Worlds Observatory, to look for signs of photosynthesis with directly reflected light off these planets. So we'd really want to gear up to help in this design. This is to my knowledge, is the first time, despite photosynthesis being such a strong and maybe the main focus of the search for life, this is the first time that there's actually been convened a workshop on photosynthesis as a producer of biosignatures. And so Bob is going to give a little bit of history on astrobiology. Yeah.
What do I do here?
Oh, we didn't think so. Sorry, forgot. I'm gonna want to also, first of all, thank the Science Organizing Committee. So could you all stand up? You're on the SOC. Thank you. So so we've been slogging away for actually some of us a little over a year now, with really early mornings in Japan and late nights in Italy, and people have had great input and bringing you all in, making sure we got your major contributions in. So thank you to the SOC. All right.
Nancy asked me to say a little bit of something about the history of the NASA exobiology and astrobiology programs. This is very sort of eclectic. It's not meant to be a complete history of this, but the 1960 the exobiology program was first started, and the first director of the program was Dick Young, who I never met, but he was quite a force in the early days. And then in the 70s, there was a lot of energy generated by the Viking missions to Mars and the putative finding of what may have been what many considered to be evidence for life. And that kind of gave the field a boost. Then in the 1980s, a lot of evidence about life in extreme environments, origin and early evolution of life, and meteorite analysis kind of propelled the field forward. And then in 1998, the Astrobiology Institute was created. Now, I came up with a few of the early pioneers in this area, and these are all people who are no longer with us. And I just already mentioned Dick Young, but of course Carl Woese was the inventor of the three-domain idea for organizing life. Lynn Margulis is the endosymbiotic pioneer. Carl Sagan needs no introduction really to anybody here. Stanley Miller, who did the early work on the Miller-Urey experiment. Dick Holland was a geologist who did a lot of work on the rise of oxygen and so on. Norm Pace also worked with Carl Woese and did a lot of work on RNA catalysis. And then the last person here may not be familiar to many of you, John Cronin, but that's more a personal choice of mine. And that when I first went to Arizona State in 1985, John Cronin was one of my colleagues there. And he and his colleagues had done some of the early analysis of meteorites, Murchison meteorite and so on, finding amino acids and so on. And John kind of mentored me and took me under his wing and took me to some of these NASA meetings and introduced me to people and really got me kind of integrated into that group of people. So I will always remember him very fondly. So, yes. Really? My bad. He would be. I always said if ever Norm Pace was in the room, everybody knew it. I thought I had seen that he had passed away. I apologize, but he went past it. He refused to submit. Good for him.
This is being recorded.
Well, Norm, if you hear this, I'm happy you're still with us.
All right. Thank you, Bob. And I just, Chris just reminded me, I guess we should introduce ourselves, especially people online who, we all know you, but maybe not everybody online does. So I'm Nancy Kyung. I'm with the NASA Goddard Institute for Space Studies, which is based in New York City, but actually sit at JPL at the Jet Propulsion Laboratory over in California. And Bob.
I'm Bob Blankenship. I'm retired.
I'm Chris Gabriel. I'm an assistant professor at the University of Wisconsin-Madison.
Okay, sorry about this. Okay, so thank you all. I'm just going to give a little bit of, Bob gave us the early history of astrobiology, and of course now we are along a long lineage of decadal strategies, or semi-decadal, that have been put out by NASA for developing the science of astrobiology and supporting it. So there have been various roadmaps along the way, and the community now is in the middle of this decadal astrobiology research and exploration strategy that's being developed. If any of you are going to the Astrobiology Science Conference next week in Madison, there will be a presentation about this, and people have been working hard on input. So there'll be more on that with other presenters here. Now we're all very interdisciplinary, and not all of you maybe have read the primers, been on the primers yet for knowing about exoplanets and stars and photosynthesis yet. So I'm going to present. There have been various frameworks introduced or developed in order to try to figure out how to put all the different disciplines together. So I'm going to present one, and apologies, Vicky. Vicky hates this setup, but I'm going to present to you. So this is something that we will all be discussing. Okay, so this is a setup that's developed by Catling et al., coming up with a Bayesian framework for assessment of biosignatures, to know whether we've found life or not. And so Bayesian framework is suitable approach when you want to come up with a probability for a hypothesis being true given very incomplete information. So we're going to have very incomplete information with sparse observations of exoplanets. So first off, the astronomers will go out trying to characterize stars, find where planets are, and various planetary system contexts. Then the next telescopes come along to try to characterize that planet a little bit more, maybe say something about its atmosphere, its size, or something. And then from those data, we will be wanting to look for any aberrant signals that look like that couldn't be abiotic, and we're going to have to be able to say, is that life or not? And also we have to figure out, could there be false positives or false negatives in interpreting that signal? And within all this, there's a kind of a framework of modeling these systems, putting all the different kinds of understanding together to simulate these planets, see if we can reproduce what we're seeing. And all that feeds into a kind of a Bayesian framework of what's the probability that we see that signal given the context, supposing there is life or supposing there is not life. So this is just a very broad framework, but it gives you maybe some idea of where your own work might fit in among other people's work. Those probabilities are only part of the equation, though. Okay, by the way, and of course, these all feed back to each other. There's a lot of iteration between these different bubbles. So the larger equation, of course, is the Bayesian whole equation, where ultimately we want to say what's the probability that we've detected life given the data and given that context. And those equations on the left in the little box, there's only part of the equations that are in the red here. But what we really need to constrain, if we can, what's the hard part, is what is that probability that life can arise, or photosynthesis, or some specific feature of life in that context. So I think a lot of your talks will maybe have some bearing on this. So in listening to everybody's talks and in thinking about your own, think about how you nestle into helping us constrain these probabilities, because claiming life is an extraordinary claim, and we want to make sure nobody jumps the gun, and these things go to the press and they go to the public, and we don't want to lose credibility by claiming something that's not there. So we present as probabilities. So with that, for this workshop, how are we going to sort of fill in some of those probabilities? So this workshop, you've been carefully curated and selected, and so we can dig deep into some fundamental limitations on and potential for aspects of photosynthesis. That includes the spectral and also intensity limits for biological light harvesting, and biosignatures of other types of photosynthetic organisms than oxygenic. Oxygenic has dominated the conversation, but we know there's a lot of other kinds there that could dominate other contexts. And also what's the geochemical context for the emergence of really critical molecules. We really haven't constrained how they originated. Also for carbon fixation and the productivity, that tells us something about how strong that signal might be for a very blurry image of a planet. And also understanding how the biosphere and planet co-evolve with each other and change through time, because the planet's going to be observed at any possible point in its coevolution with the biosphere. And then how what will we actually be able to observe with upcoming telescope missions of these exoplanets. And from this workshop, we're hoping to produce, you're all going to help contribute to a white paper that will help inform this NASA DARES strategy development. But we want to help really constrain our confidence in finding and detecting photosynthesis on an observed planet. And hopefully, when you come away from this workshop, your research will be further inspired with new ideas and also cross-disciplinary collaborations. So that's our goals. And without further ado, let's get into the practical matters. Chris, take it away.
Okay. This is quite an angle here. So I think you all have schedules. If you don't have a paper copy of the schedule and you want one, just front and back, there's a big stack that's out here on the table. There are also versions you can download online, so please grab that. Just a few things I want to point out about the schedule. On the top there, be aware that tomorrow the breakfast is relatively early, so just be aware of that. And then also we have recreation time, 2:30 to 6 p.m. tomorrow. I'll tell you some things you might want to do for that. And then be very aware, if you're taking the bus back to Madison, that's going to leave at 1:30 p.m. on Sunday, so don't miss that.
Which might say the session starts at 8:30 tomorrow instead of...
Yes, and the session starts at 8:30 tomorrow, which is earlier as well.
Can you say something about the breakouts and working?
What do you want to say about that?
Okay, so we are going to have breakout discussions. Every day there will be a session. So the first one, we're just going to have our chance to blab and ask them questions and find out who each other is and what we can find out. But then and we will also be trying to brainstorm topics for what's going to go into the white paper. And also brainstorm. Also, if you think there should be some review papers on some topics, we might develop those from the workshop. So then the working groups will get into actually organizing and finding leads for the papers and developing a plan for putting these together.
So here's a few things. I have done absolutely none of these things, but here's a few things that you could do in this area. These are, I might describe these as hikes, but here in the Midwest, they're just flat walking paths, right? So here's a few that I can suggest. Notice that 3 of these are around 15 minutes away. So there's some of us that have vehicles that maybe you can tag along with. I have been told that you can take Ubers, shorter distances like this, probably not to Madison or Milwaukee, but also the Green Lake Marina is relatively close by. However, it would be about a 30-minute walk, so it's only about a 5-minute drive. So consider those things if you would like. There are a few other Native American, attraction sites, that sort of thing. If you'd like more info about that, just let me know and I'll give you that. And then lastly, just be aware of this code of conduct. We always want to be respectful to one another while having conversations. I think a huge part of this workshop is being able to have open conversations, address disagreements, things like that, right? We're going to have a lot of discussion. So please practice active listening, constructive critique, no harassment, obviously. Please do not do anything like that. We want to be welcome back at this place. Be very inclusive. Please, be aware of that when you speak, and also be aware of anything that is confidential, right? And credit those where that's needed. And then lastly, the tech etiquette. If you're online, please be sure to mute yourself, whether you're in a room or somewhere else, and just be aware of others. And if we ask you to do something online, please cooperate and pay attention as well. I think that's it for code of conduct. Anything else? Okay. Good. For the speakers, be aware that there are also lapel mics if those are wanted. Okay.
I'll open her screen. I'll open her presentation. Vicky.
Putting this here is probably good enough.
Where's Vicky? Oh, there she is. Okay. All right. Share. It's sharing. Okay, let me stop sharing this. Okay.
It was already here.
There we are.
Yeah. Okay. We're here for the first session of the day. My name is Avi Mandel. I'm from NASA Goddard, outside of DC. I will be chairing this first session. I'll let Vicky start to get the lapel mic working.
I think, is it special? Yeah. Can you hear me?
I think so.
See me, you can hear me. Excellent.
Yeah. So little introduction, though Vicky doesn't need much introduction. Vicky Meadows from the University of Washington, SETI Institute. Also the PI of the Virtual Planetary Laboratory. And a very pertinent thing for this discussion is that she's also on the Community Science and Instrument team, the CSIT, which you might hear that acronym thrown around, the CSIT group for the Habitable Worlds Observatory, which is primarily tasked with helping to define the level 1 requirements and the driving science cases for the upcoming Habitable Worlds Observatory mission concept, which you might hear more about from Vicky. So hopefully we'll have about 30 to 40 minutes of speaking and then questions afterwards. And then we'll have 1, 2, 3, 4, 5 other speakers in the session with a break in between. So, all right, thanks. Take it away, Vicky.
Okay, great. Well, thank you to the organizers. This is extra mic going to cause a problem. Yeah. Yeah, there we go. All right. Thank you so much to the organizers for inviting me to come and give this overview. I do have a lot to cover. Nancy gave me a list of things, and so I'm going to do my best, and we'll see how we go with this. But what I'm going to do is going to give you an overview of, from an astronomer's perspective, what we are. Oops, what's going on?
Oh, we're gonna share your video.
Oh, okay.
Okay, sorry about this.
From an astronomer's perspective, I'm going to tell you about, what we're thinking about as far as detecting photosynthesis on other planets, kind of planets we might look for, the techniques we're going to use to try and search for life, the sorts of things we're thinking about looking for now. And I'll finish up with sort of big gaps that we need filled, which hopefully people in this room can help us with. All right. So, the question when I say that, I search for life on other planets, that's what my science area is people say, well, how do we search for signs of life on other planets? Because no matter how good our telescopes are, we're never going to be able to resolve that pale blue dot there, which is the Earth seen from the Cassini spacecraft. We're never going to be able to resolve that into an entire world. And so we're trying to understand life from this pale blue dot. And so in the spirit of what Bob was doing, if we look back into the history of the field of astrobiology, there were two sort of main ideas about how we might actually go about looking for signs of life, and in fact, photosynthetic life on planets around other stars. And the first paper, I love this paper by Hitchcock and Lovelock, which really goes through, how do we detect essentially a photosynthetic biosphere in the atmosphere of our world? And so there's very much, looking at this concept of a disequilibrium biosignature. But if you go through this paper, it also talks about false positives. It talks about a lot of the concepts that we're now sort of grasping today. And then we also have this very early paper by Gates et al. Looking at the spectral properties of plants. And in one of their papers, they actually speculate, we could maybe see this from space too. So, so these two things, looking for how life impacts the atmosphere of a planet, and then how life affects the spectral reflectivity off the surface of a planet, are sort of two of the key themes that we'll be exploring here. So how do we pick the best planets to search for life? Well, there's kind of a consensus at the moment, because we have to start somewhere. We're going to go and look in this region called the habitable zone. So this is a region around a star where an Earth-like planet could maintain liquid water on its surface. But the interesting thing is, if you delve into that definition, Earth-like planet isn't quite what you expect. Okay, but first of all, if we look at on face value, Earth-like planet, okay, what are we looking for? So we're looking for probably an Earth-sized world, one with a rocky composition that could hold an ocean on its surface. And we now know enough from exoplanet population statistics and the ability to get both radial velocity and transit measurements for stars, for planets, that we're able to actually figure out the density of these things and figure out that, yes, actually, things below about 1.5 Earth radii are truly rocky. So we'll be looking for those. We'll be looking for them in this habitable zone here, which is this region around a star with, the fraction of starlight relative to what the Earth would get is along the bottom there. And then along the y-axis is, the different type of star that you're around. And you can see that the habitable zone actually moves outward if you're around a smaller, redder star there at the bottom. And that has to do with just where the star's light's coming out and whether or not the planet is able to absorb that light and heat up with actually less starlight to be more efficiently heated. So that pushes that out a bit. Okay, so we have a ton of things. You can see them all lurking around the bottom there. We have a ton of potential planets that are potentially habitable in the habitable zone already. But I want to focus on the outer limit of the habitable zone, because actually it tells us something really interesting, and it will be relevant to photosynthesis. And one is that outer limit is called the maximum greenhouse limit. It occurs, we believe, when a planet has so much carbon dioxide on it that if you keep adding carbon dioxide, it actually ends up cooling the planet because of the scattering from the atmosphere of the carbon dioxide. And so this was actually calculated in a very, very, classic paper by Ravi Rapu et al. On the habitable zone. And basically they showed that once you add about 7 bars of CO2 to a planet, if you keep adding CO2, instead of having greenhouse warming from the CO2, you end up cooling the planet. So that is the outer limit of the habitable zone, the point at which you can't get the greenhouse to work any further. And interestingly, on this diagram, you can see the 2 circles for Earth and for TRAPPIST-1e down the bottom there, little planet right on the x-axis there. TRAPPIST-1e is a little further into the habitable zone. It's further away from its star. It actually needs more CO2 to be habitable. It needs about 5% CO2 in the atmosphere compared to our current WINS's 431 ppm that we currently have in the atmosphere. So that's interesting. So you think, okay, if a planet has to be habitable at the very outer edge, and you need more as you move into the habitable zone. What does it mean to be an Earth-like planet in the habitable zone? And so the habitable zone concept, the actual extent of that habitable zone, assumes an active carbonate silicate cycle with a negative feedback between temperature and atmospheric CO2. So that's this thing for all of the geologists in the audience. I thought we might put one of these up. And really, this kind of breaks down to a cycle, which basically says if you increase the surface temperature on a habitable planet with active volcanism and an ocean, the temperature will rise. That will cause rainfall and silicate weathering rate to increase. That will draw down the CO2, which will reduce the greenhouse effect, which will then reduce the surface temperature. So by increasing the surface temperature, you activate a negative feedback loop. Same the other way. You reduce the temperature, you reduce the weathering, you build the CO2 up in the atmosphere, and you increase the greenhouse effect. So when we say an Earth-like planet in the habitable zone, we mean one that has an active carbonate silicate cycle. So as you move through the habitable zone, your planet is going to get more and more carbon dioxide as it heads towards that outer edge of the habitable zone. And this is a lovely diagram that Owen Lemur worked on, actually showing the amount of CO2 a planet would have to have. And so we've got the abundance of CO2 there on the y-axis. How much CO2 this planet would have to have in its atmosphere to be able to remain habitable throughout. So remember that. An Earth-like planet in the habitable zone could have massive amounts of CO2 towards the outer edge, and in fact is required to have massive amounts of CO2 for this to work. So as photosynthesis experts, you're maybe thinking that's interesting, and we'll come back to that, right? Okay, so how do we study exoplanet environments? We have two main techniques. One is using a technique called transmission spectroscopy, and that's where, just in this lovely diagram taken from the space shuttle, you can see the sun behind the Earth's atmosphere. You can see the glow as that sunlight passes through the atmosphere. I love this figure, though, because it also shows how incredibly hard this is. We're going to try to do this for a planet transiting in front of its star. So as a planet passes in front of its star, and we found plenty that are actually in the right orientation, we'll get that backlit atmosphere. And because the atmosphere is able to absorb radiation, what we will end up seeing is that more of the star's light will be blocked out by that planet at wavelengths where molecules are absorbing. So where no molecules are absorbing, you only get the fraction of the starlight blocked out by the solid body of the planet. But if you're, say, in a water band, then essentially you get part of the atmosphere is opaque because of the water band. And so the planet appears to get bigger. And so in these diagrams, what you're actually seeing is the effect of a smaller planet versus a bigger planet. Where the planet is bigger, the atmosphere is absorbing. And so you're able to see the actual structure of the molecules there. And I want to point that out because when I first entered this field, I was completely confused by the fact that absorption is usually the downward thing, right? Not the upward sticky thing. In transmission spectroscopy, your absorption is in fact a mountain, not a valley, as we would expect. So because we can do this, we can actually use these transmission spectroscopy observations to probe the upper atmosphere and stratosphere of habitable zone planets. It's not great at getting down to the near surface environment, and we can't say anything about the surface itself because we skim along the top of it. And so it can give us some important information. A better technique, though, is the one that will be used by the Habitable Worlds Observatory, which is to be able to directly image the actual planetary system. And so here you can see this lovely movie of sort of planets orbiting. This is a solar system analog. Planets orbiting around that central star. Because we're able to separate the star from the planets, we can then isolate those planets and get spectra of them. So, you know, in this diagram, you hone in there and you can put a spectrometer essentially on the dot that is the Earth here. And you're then able to take the reflected light spectrum. So this isn't transmission. This is light from the star hitting the planet, being reflected off the planet, coming back to your telescope, and you're able to observe that. So that reflected light spectrum goes, if there's not total cloud cover, it goes all the way down to the surface and comes all the way back. So this technique can tell you something about the surface of the planet. It can tell you something about the near surface environment. And it may even let you, in fact, detect oceans. So I'm just going to point out here that it's a sort of bunch of features in this spectrum. This is just a squiggle, but the squiggle means a lot to spectroscopists, and it shows key elements that we care about, things like abundant oxygen in the atmosphere, water vapor bands, and then, of course, our methane, which is probably not cows, it's probably methanogens, but anyway, we always put a cow. Okay, and it's Wisconsin, so why not? So, okay, so again, potentially, if we can see all the way down to the surface in direct imaging, then we can start to learn something about what's actually on the surface. And this experiment is a really lovely one. It's from the EPOXI spacecraft, which was in the solar system. It is, in fact, the Deep Impact spacecraft under a different name. And what happened was once it finished its primary mission, it made the mistake of continuing to function. And so it was then used to look back at the Earth and to take these light curves, which are just photometry. So we're just measuring the brightness of the planet. Here with epoxy, we can actually see the details on the planet, but imagine for an extrasolar planet that you only just get sort of a disk average of each of those. But if you look carefully, you can see on the blue one at the very end, right near my name for some, I'm not sure why my name is there. You can see that it goes down. And then if you could look in the infrared image at the bottom here, you can see that it's going up at the end. So the color of the planet is changing as it rotates underneath us. And we can in fact map that color change. So you can go to something called an eigenspectrum where you pull out components that can be co-added to make that color change. And then eventually that allows you to actually pull out that longitudinal map on the bottom, the thing that looks like the beach ball. And that is at least hinting at the presence of continents and oceans in this particular case. So even though we can't resolve that pale blue dot into this lovely image of planets and oceans, we could potentially map it to see something about the surface. Okay, so I'm going to talk just very briefly about how we might search for signs of habitability, because that also falls into how we assess whether or not we're looking at a biosignature. So direct imaging spectroscopy could also potentially give us planetary surface pressure and temperature. Because if we're potentially getting down to the surface, we can use a technique called retrieval. And it's kind of exactly what it sounds like, is we go into the data and we retrieve, things about the environment from that spectrum. So planetary surface temperature and pressure can be extracted from the spectrum via this process called retrieval. And so that could tell us, it could say, well, the temperature at the surface is this and the pressure of the atmosphere is this. And if we then take that temperature and pressure and you plot it on a water phase diagram, we can tell whether the temperature and pressure that we're observing is actually falling into, is liquid water possible here, or is everything too hot and it's gas, or is everything frozen and it's solid ice? So this particular plot here is actually done by my student. She was modeling a whole bunch of different atmospheres for the TRAPPIST-1 planets, and we found that some of them actually do fall in the liquid water zone that are feasible atmospheres that they could have. But again, this is not backed up by data. This is just theory at the moment, but it's kind of illustrating, temperature and pressure, we could maybe do that. The other one is a far more, I think, romantic, but unfortunately more challenging way of doing this, which is to try and detect the actual glint of sunlight off of an ocean. So people have called the habitable zone the light on water zone, which I think is a lovely poetic way of talking about what we're trying to do here. So maybe we could, in fact, see light reflected off of water, and that would give it away. So in our own solar system, you can see Titan there. You see the bright spot on Titan's crescent. That's actually a reflection from Kraken Mare, which is an ethane lake. But here for the Earth, in this shot here, these are images of the Earth taken from the L-Cross spacecraft as it did its multiple swings around the Earth and the moon before it crashed into the moon. That was its mission. It was supposed to crash into the moon. So that was successful. But you can see, if you look at the Earth through its different phases, near crescent phase, you can see that bright dot on crescent phase there that I've circled with the red circle. If you can see that, then you are a remote sensing ocean detector. Okay, there is the glint from the Earth's ocean being seen very strongly in the crescent. So if we can take observations of an exoplanet at different phases, so when it's half illuminated versus much closer to crescent phase, we can actually see the difference in the reflectivity of those surfaces. And this was work done by my then graduate student, Jake Lusty-Geiger, looking at actually mapping the planet, as I showed you, taking out the surfaces that were potentially continent and ocean, and then plotting them. So the continent surfaces, the orange lines there, you can see the two lines of the two different phases. You can see at the two different phases, the orange lines pretty much fall on top of each other. But at the two different phases, the blue lines are massively different. And so that water surface becomes much more reflecting at crescent phase than it does closer to dichotomy, to half illuminated there. So that would be a giveaway, and that would help us detect whether or not an ocean is there. Okay, so the meat of this, how do we search for signs of life? Okay, so we are looking for biosignatures, and in the exoplanet sense, biosignatures are the potentially detectable impact, and it's often primitive life, of primitive life on a planet's environment. So when we talk about looking for different types of biosignatures, these are sort of the three main categories that I think about. One is targeted biosignatures, where we hypothesize a metabolism and we say, okay, we know what the products of this metabolism are, like, let's say, photosynthesis. So we're going to go after these particular things. And so we have metabolism in mind. We have a process in mind. It allows us to hone our search to particular things that we might want to look for. So that could be gases in the atmosphere. It could be the red edge reflectivity of plants, things that are temporally varying, or things like pigments or fluorescence from our pigments as well. And there's a paper there by Yukamatsu, who I'll call out because he's actually here. It's over there. Okay, and so the other thing we could then look for are agnostic biosignatures. There we don't assume any metabolism. We simply look for anomalies in the planet's environment. We look for not planet. Okay, so our life detection is not planet. So what we're trying to do is discriminate the things that we think are normal planetary processes, photochemistry, volcanism, etc., and find an anomalous process working in the environment. And then, of course, technosignatures. Technosignatures are kind of the ultimate impact of life on its planetary environment, and that's something we could potentially look for as well. Oh, yes, and for an excellent review of the diversity of biosignatures, you can see a review that Eddie did from a previous biosignatures workshop we had, and Eddie's here as well somewhere. Oh, there he is. Sorry, you're right in front of me. Good to see you. Looking down. All right, so different types of biosignatures. Right, there's the classic ones. There's the disequilibrium you've probably heard of, which is oxygen and methane simultaneously together. That's really, it's really kind of more of a stronger emphasis of methane being a biosignature in this particular case, but also the oxygen also. It does require that you evolve both methanogenesis and oxygenic photosynthesis on your planet. So maybe that's not one that we necessarily see commonly, but it's something that we go after. This grainy plot here is actually from a fabulous experiment done by Carl Sagan and his colleagues, where they ran the Galileo spacecraft past the Earth and one of its gravity assists. They begged the engineers to turn the instruments on, which they did, and they studied the Earth as if it was an extrasolar planet. And so part of that is they're looking at, you can see at the far column there, thermodynamically, here's what you expect from methane, nitrous oxide, and ozone, for example, in the environment, and yet the actual measurement they got, the Galileo value, is way higher, implying that there is in fact a surface flux of gases, of these gases in the environment, or in ozone's case, a conversion. Another way we can also go after biosignatures, not just the oxygen methane pair, but oxygen, nitrogen, and the presence of an ocean. So remember, trying to see that glint and see that ocean. That's also potentially a biosignature, because kind of the equilibrium state would be a nitrate-rich ocean. But instead, we have biological mediation to put that nitrogen and oxygen back in the environment and in the atmosphere above. Ozone can also be used as a nonlinear proxy for oxygen. What I mean by that is if you have abundant oxygen in the atmosphere, you will photochemically produce ozone from that, but not really in a linear fashion. And so you can see this lovely classic plot from Cassidy and Donahue back in 1980 that actually shows, as a function of PAL is the present atmospheric level of O2, so that's the 20 to 21%. As a function of fraction of the present atmospheric level, you can see the kind of ozone column that you get. And eventually, of course, that curves over and saturates at higher abundances. Probably self-shielding, I would guess. And there's also been a lot of work by Thea Kazakis as well on sort of looking at what that means for the LIFE mission also. So there's a series of papers on ozone as a biosignature. And then I just want to say that and this is kind of a key figure from the LUVOIR final report. It was in the Astrophysics Decadal as figure 1-1, which was just so exciting when I first opened that document. It's like, wow, first of all, it is work done by the VPL, but it was put together in this diagram for LUVOIR. But this really is sort of the main case that was made and allowed us to justify the selection of this mission, was that if we can get spectra of exoplanets, even throughout the entire history of our planet, we should be able to pick up biosignatures at each particular stage of its development, from the methanogens in the Archean to potentially the presence of weak ozone, which you can see in the ultraviolet on the extreme left-hand side of that middle plot, spectrum plot there, that's the ozone being formed from the oxygen. Even though the oxygen is very weak and you can't see it in the spectrum there, the ozone may be detectable. And then here in the modern day Earth, when we have our vegetation and our cyanobacteria working for billions of years, We have the oxygen, we have the ozone, and we can potentially also get methane as well. Okay, but what if I can't see oxygen? Well, methyl chloride is another potential gas that could be produced. So in this particular work I'm citing here, earth scientists essentially looked at the emission of methyl chloride from both coastal areas and also tried to pin it down to being produced by tropical plants and ferns, which they were able to show that tropical plants and ferns actually also give off methyl chloride. So that's kind of a secondary indicator of that. Okay, another thing we could potentially look for, surface reflectivity signals. Different types of pigments will absorb, and then potentially the planet will reflect beyond that pigment. Once it's got what it needs, the pigment wavelengths, it becomes reflective beyond that. But here's actually more work by Eddie showing that in fact that rise and where the pigment actually is obviously a function of the organism itself. And in this case, we're looking at halophiles with different pigment positions. It wouldn't be a photosynthesis conference if I didn't show Nancy's wonderful paper from 2007, which kicked off a lot of this, where we looked at the coevolution of the photosynthetic mechanism essentially with its environment. So looking at, where am I going to evolve my pigment, or where am I going to do my absorption, essentially, given the spectrum of my star and the absorption of the atmosphere that is above my plant or my photosynthetic organism, where in this spectrum is it optimal? Where do the photons come down, being peaked from the star and dodging all these different absorption bands within the atmosphere? And so we were able to show, there's quite a difference between sort of the peak radiation from the star hitting the top of the atmosphere in that sort of green diagram at the top there. You can't see my point. Oh, can you see? Oh, you can see my pointer. Fantastic. Okay, so up here at the top of the atmosphere, and then by the time you get to the surface, it's a very different distribution. The peak is in fact shifted much closer to where we normally expect the chlorophyll A band to be. Temporal variability is another thing we can look for. So looking for variability in CO2, which is actually due to the annual drawdown and release of carbon dioxide from photosynthesizers on our planet. And then also potentially looking, when oxygen has not risen to be highly abundant in the atmosphere, looking for the variation in that UV ozone band. So we can't detect the oxygen, it's too weak, but the ozone has a very strong band in the ultraviolet, and we can see that actually going through seasonal cycles as a part of that process. Again, a lot of what I'm showing is theory, and that UV model is definitely theory. The Keeling curve is real, okay? So, and we have to deal with that. Okay, so we have all of these sort of postulated ideas about what we might actually get to see, but the host star can enhance, suppress, or mimic biosignatures, and we have to keep that in mind. And so this is a sort of a classic example between the green and the blue spectra there. Even if you don't understand spectra, you can see that those two are very different. And that's because one of them is an Earth-like planet orbiting around the sun, and that is the blue line. And the other one is an Earth-like planet orbiting around Proxima Centauri, which is an M dwarf. And what you're seeing is a huge enhancement in the lifetime of methane for the M dwarf planet, and a suppression of ozone for the M dwarf planet. And that has to just do with the photochemistry. The ultraviolet spectrum of the star drives different lifetimes and different, therefore, abundances for the same surface flux. So if we have the same surface flux of methane coming off the planet, you just build up more of it on a planet orbiting an M dwarf. So we need to take these sorts of things into account when we're trying to interpret what we may actually see. And that also leads us to, so that's enhancing and suppressing things. And then the other thing is mimicking things. And so this diagram is kind of a summary of a bunch of work done by a lot of people. And you can see the citations on the papers there, where people realized, oh, I can actually generate oxygen using a photosynthetic process, using a photochemical process in a bunch of different ways. And so this was, the famous story was that I was putting the annual report for VPL together, which is not always a hugely fun task, but I was putting all of this stuff together, and I just, as I gathered like things and put them in a paragraph, I realized, oh my goodness, we have all of these different techniques. And so that is something that I think, Nancy asked me to highlight some VPL stuff. I think that's something we'll be remembered for, is just pointing out that even though oxygen was often considered the slam dunk biosignature for life, Certainly, some of the very early missions assumed that. We found that, yeah, actually you can get false positives for this. So in different environments where there's not a lot of water vapor, or you have in fact lost an ocean and water vapor has evaporated, fertilized, hydrogen's escaped to space, and you build up massive amounts of oxygen, there's sorts of different scenarios in which oxygen will build up without photosynthesis. But on the other hand, there's also tells in the environment. There's things about that environment that don't look like a sort of thriving photosynthetic biosphere, but then might tell us, oh, this looks like a post-ocean loss planet, or this looks like a planet with almost zero water vapor and therefore, stable CO2 photolysis. So people came to me and said, this is tragic, this is terrible, we've lost our gold standard biosignature. But I think instead it actually matured the field, because it told us that we can't just assume that something is a biosignature. We need to also look for its potential false positives in the environment. And every single one we rule out strengthens our conclusion in our statistical framework that this is in fact due to life. And speaking of statistical frameworks, I don't really hate statistical frameworks. I actually love statistical frameworks. I just don't like the ones that insist I have to prove that the planet was able to support life in the first place, because I'm an observer and I'm like, hey, there's an environment right under me now. I'm getting a spectrum. Give me the probability that, given the data I have now, there is life in this environment. And so the community, about 500 people in total, got together to draft, to review, to have comments on a framework for assessing biosignatures. And this was considered to be sort of a universal framework, in the sense that these 5 questions are so general they can be applied to both in situ biosignatures, so biosignatures in rocks, for example, and remote sensing biosignatures as well. So this is not just for exoplanets. We have these 5 fundamental questions, which is, first of all, have you detected an authentic signal, right? If you're claiming that you found life, have you actually found something on the planet, or is this just a reflection in your instrument, right? Have you properly identified the signal? Have you identified it as the right molecule, for example, or is your spectral resolution so low that it could be other molecules and you have to rule them out? Are there abiotic sources for your detection? So that's the false positives thing. It's like, okay, so you detected oxygen in the atmosphere. Can you rule out all these other false positives? Is it likely that life would produce this expression in this environment? That's another one that has to be done in parallel with the false positives, because sometimes you can claim that life is doing something in an environment, and yet when you look at the thermodynamics of it, it just doesn't make any sense, for example. So these are the sorts of things that you could look for. And then question 5 really is, okay, you have your life and your non-life hypothesis. Are there additional measurements you can make that can help you discriminate between your life and your non-life hypothesis for the explanation of the signal that you see? And we have this lovely diagram kind of showing the process in 2 different stages. You want to determine your data is correct and you're actually detecting something before you release the rest of the community on to try to figure out what's actually going on. And that was put together for us by Mike Wong, who is also in the audience here. Okay, so in the final stage here, how are we actually going to do this with the instrumentation that we have planned? So there are 2 paths for searching for life on exoplanets at the moment, which were first delineated in the 2018 Exoplanet Science Strategy. The first one is called the M-dwarf opportunity. So right now, we have both James Webb Space Telescope and the ground-based ELTs will be coming online before the end of the decade, maybe for the ELT. I'm not sure what the final schedule on that one is. They will let us do this transmission spectroscopy thing on a limited number of M-dwarf targets. And so we'll be able to start to look for life with those. In the longer term, we have missions like the Habitable Worlds Observatory, and which is a go. It was selected in the decadal survey, and so we are actively working on it right now. And we also have the Life mission concept from Europe, which it's still a concept at the moment, right? So that's going to go up for competition to be selected. So those are coming, and the M-dwarfs are transmission. HWO and Life will be doing direct imaging by various techniques. So if we look briefly at the M-dwarfs, and I apologize profusely to my European colleagues, it says US ELT here because these slides, and I was reviewing it last night and I was so tired and I didn't pick it up, but the US ELT was because these slides came from an actual political presentation that was essentially trying to convince the NSF to continue funding these things. So that's why I emphasize US in here, but also, of course, the European ELT is very much part of this landscape. So JWST and the ELTs coming down, able to detect signals on M-dwarf planets. I will point out, though, that when you're doing transmission spectroscopy with JWST, there's only really a handful of planets on which you can do transmission spectroscopy. And that's because of this effect here. This effect is an Earth-like planet put around AD Leo, an M3.5, I think, dwarf star. So it's a fairly large dwarf star, as large dwarf stars go. And then we've got Proxima Centauri and TRAPPIST-1, which are smaller and smaller stars. And as you put a planet around these smaller stars, remember, you're trying to block out a fraction of the star's light to see the size of the planet change. If that star is smaller, then you block out a larger fraction of the star's light with the atmosphere than you do if the star is larger. And so because of that, you'll end up getting bigger atmospheric signals. So for that reason, we're trying to detect life on planets around really small M dwarfs, just because that is just the easiest way to, in fact, get that detection. So my group and others have done a bunch of simulations on, okay, can we find life with JWST? So TRAPPIST-1e in the habitable zone of the TRAPPIST-1 star is considered to be the most promising planet. It's an Earth-sized planet. It's in the habitable zone. The question is, could we actually detect, for example, oxygen in the atmosphere of this planet if it was there? You can see my little, sign up there with oxygen pointing to the oxygen bands, these little green things here. They are tiny at the spectral resolution because they're very narrow features. And so in the near infrared to mid infrared, it's actually extremely challenging to observe. Well, there's no mid infrared signals of oxygen, but there is a mid infrared signal of ozone, but that's also potentially challenging. This is 100 transits with MIRI LRS, so even that would be extremely difficult. Anything beyond 10 transits is a bit too much to ask for telescopes. So yeah, and it's not just us. There's a whole slew of people, multiple groups have independently found that it's highly unlikely that we would see photosynthetic oxygen with JWST, which is depressing, but I'll give you something better later on. And so we couldn't detect photosynthetic oxygen, but we might be able to detect if the planet's lost in ocean, though. You can see these massive features, which are collisionally induced absorption from oxygen, crashing together in a 3-bar atmosphere that is post-ocean loss. And so who knows, we might actually detect oxygen, but it won't be photosynthetic oxygen. We could also potentially look for fractionation signals from HCO as well in the atmosphere. So we might be able to see a post-ocean loss planet, but the photosynthetic one, not quite so much. JWST will also struggle with methyl chloride, at least from sort of the tropical plant kind of idea. And it might look like you're kind of seeing something there, but again, the noise from the instrument is such that it's going to be extremely challenging. Eddie and his group with his student Michaela Leung actually looked at microbial methyl chloride as well. And Michaela argued that you could actually, if you pair that with methyl bromide, it might in fact be detectable in 100 transits with MIRI, but you need about 1,000 times flux than we have on our own planet. So that is going to be challenging, maybe unlikely to see a planet with that level of productivity. Okay, the ELTs, though, will allow us to do oxygen. Okay, so we can't do oxygen with JWST. We can do it with the ELTs. And so in this particular technique, they use both high-resolution imaging to be able to actually isolate the planet from the star, to be able to get an optical fiber on it, to run it to a high-resolution spectrograph. That high-resolution spectrograph, you can see there, the sort of the scared cat look, that is breaking the oxygen band up into all of its constituent lines, which is superb and amazing. And because the planet is moving through space and its star is moving through space, those lines will be shifted relative to the telluric absorption lines, the lines from our own atmosphere. And so that allows us to potentially isolate the oxygen on the planet. So that is a technique that's coming up that could potentially work. I'm going to move a little bit here just to wrap up. My then student, Miles Curry, looked at sort of detectability using transmission. We found that oxygen in transmission would be a little bit difficult, 100 hours of observing time. But the methane and CO2 biosignature would leap out. That's not from photosynthesis, but we still have to love all the biosignatures. But he also looked at sort of direct imaging. So the interesting thing with the ELTs is we can do two techniques. One is the actual transmission. If we have a planet that transits in front of its star, we can do that high resolution spectroscopy. But, and this is great, we can also use reflected light even though we don't resolve the planet. okay, because of that relative motion. We can break that out. So if we use the reflected light technique instead on planets that are potentially closer to us, like Proxima Centauri b, for example, then that makes the oxygen potentially more accessible. So that would be kind of a key experiment, would be looking for oxygen on the planet that's orbiting around the closest star to our solar system. So that's coming up. I'm sure that experiment will be hotly sought after by multiple people. Okay, so finishing up. So there are two paths for searching for life. As I said, there's the transmission one with the JWST and the ELTs, and then we have the direct imaging one done by HWO and LIFE. And that one will look for planets orbiting around more sun-like stars. So our star is a G-dwarf. There's no way JWST or even the ELTs can look for planets around G-dwarfs, but they will be able to do it for planets, for the LIFE and HWO will be able to get into that space. And I just want to point out that we as astronomers are deadly serious about this. So this is in our 2020 decadal. So inspired by a vision of searching for signatures of life in planets outside of our solar system, we therefore justify our next astrophysics flagship. Okay, and so that is just, this is the first time we've had I think on pretty much any astronomy mission that astrobiology has been the driving requirement for the telescope. We have a requirement to get spectra of at least 25 habitable zone terrestrial planets. Okay, so this is the machine where, well, okay, this is a preliminary architectural option, as you'll see down the bottom there, which I'm sure our engineers told us we had to put in there. But this is an artist's concept of what the telescope will look like. So this is the Habitable Worlds Observatory. It's a large telescope in space. We build a lot of those. One of its main advantages is it will allow us to search around hundreds of stars. We have a requirement to get 25 candidates that we then take spectra of, but it will allow us to search around hundreds and hundreds of stars, which means if we don't find something, it's actually statistically significant. So it gives us that. Again, it will allow us to get these spectra. But I want to point out in my last minute or so here is that at the moment, the case for this has been driven by this Earth through time concept, which is a fantastic place to start. But I will point out that the Earth is right at the inner edge of the habitable zone, where we have relatively little CO2. We need relatively little CO2 to stay warm. But the rest of the planets that we find are not all going to be right up against the inner edge of the habitable zone. They're going to be through the habitable zone. And as I explained to you, that means they'll have relatively large amounts of CO2 in their atmosphere. And so some of the experiments we've been doing, the VPL, hot off the press, I think I got this out for 2 meetings past, which was the CSIP meeting, which was 3 weeks ago, looking at what these planets look like as we put them through the habitable zone and they have appropriate amounts of CO2 in their atmosphere, and developing self-consistent results from that. And what we see is that, the CO2 bands, actually, I'll just point these out here, actually become, of course, much stronger, which is obviously what you would expect, but that has important ramifications for the design of the telescope. We also see very different abundances in water vapor. But one of the sort of really interesting things, I think, is we looked at the very outer edge of the habitable zone and said, can you actually distinguish an inhabited high CO2 outer habitable zone planet from, say, a Venus-like planet in the habitable zone, which can occur, especially around M dwarfs? And the answer is yes, you can, but it's mostly actually from the methane and water vapor as well. So we're working on this at the moment. And so this is sort of kind of driving our thinking, which is that for a long time for this mission, we thought about the thing at the inner edge, but now we have to think about everything else throughout the habitable zone. And so my key takeaways then are that astronomers are really serious about looking for signs of life, including photosynthetic life on other planets. And our next NASA astrophysics flagship is driven by it. So we really need all of you as a community. And that's my next point. The search for habitability and photosynthetic life on exoplanets is massively interdisciplinary and will require combined observations from multiple telescopes and planning interpretation from a broad community of scientists such as yourselves. So for me, the key outstanding questions are, can we address what photosynthesis will be like throughout the habitable zone? Okay, so what are the likely implications for photosynthetic processes, pigments, and productivity at high atmospheric CO2 and or at lower light levels as we move further away from the star? Can we predict evolution with the environment and pigment to get the spectral locations? So we kind of maybe have a better, either know where to look or could at least make a better probabilistic argument for the fact that that probably is a pigment, given that it makes sense in an evolutionary sense. And are there upper limits for methyl chloride production as well? That would be a good thing to have. And given the difficulty in observing O2 on exoplanets in the near term, are there any other secondary gases or other forms of photosynthesis, et cetera, that we should be looking for? So I will finish up there.
Right, that's good to see you again. We can just do this thing with the microphone stuff. So, Chris, we're going to do questions online through the owls as well as questions in the room. Is that right?
Can people type?
Oh,
Yeah.
questions in the
Yeah.
chat. Okay, all right. So folks online, please post questions. We're getting lots of hands online, which is great. Post questions on the chat. But first in the room, questions for Vicky. We have about 7 or 8 minutes for questions and discussion.
At the beginning, you said that it's more probable to find the terrestrial Earth if the radius is not more than 1.5. But then and then later on you said this is more easy to observe. So, what is the reason why you think is more the probability to have a terrestrial type of a planet when the size is similar to Earth? What is this base?
The basis for that, and again, it's probabilistically, there is a curve. So you could get terrestrial planets, rocky planets that are larger than 1.5, but the basis is actual empirical measurements. So we're able to see Earth-sized planets around the other stars and get both their masses and their sizes, so we can determine their density, and we see that they are rocky. And so you can see this distribution of 2 sets of planets, one rocky set below about 1.5, and another more gaseous, water-dominated set above that radius. So, yeah.
Well, let me just add, since as the planet's mass gets larger, we expect it to be accreting and holding on to more of its atmosphere from the initial planetary nebula. So naturally, just from physics, we expect that larger planets will have larger and much denser atmospheres, and eventually won't be, quote, rocky. They may have a rocky interior, but they'll have a huge amount of gas on top of that. Question for over.
Hello, thanks for the great talk. This is completely not my field. I wanted to ask, this was a lot of focus on kind of light harvesting for synthetic forms of life. And you mentioned false negatives. So kind of given the diversity that we have on Earth, what do we expect? What are we missing if we're only looking for light-based life forms?
So you're saying if we focus only on metabolisms that we know, what do we potentially miss? So I mean, of course, we can't know that. And then part of the activity here, I think, is to think beyond sort of classical photosynthetic mechanisms. But that's where the agnostic biosignatures come in. That's where we try and characterize the environment and look for anomalous processes that are working, but not any preconceived process, but to say there is something operating in this environment that it is doing light harvesting, or it's doing, whatever. I might be able to see that from seasonality. I might be able to see that its productivity changes as a function of how much sunlight is hitting it, for example. But that would be agnostic. We will not potentially be looking for a targeted thing, but we will see something anomalous in the environment. And so that will allow us to capture life as we don't know it, essentially.
Cool, thanks.
Do we have any questions online? Anyone who's monitoring the chat? I can't actually see it here. No, okay. Question over here.
Yeah, this is also not really my field, but one okay, yeah, that's true. Yeah, one question I have, this is also not really my field, is there any opportunity or currently to also resolve isotopic differences of the CO2 or O2, or is it something that can happen in the future now?
I mean, possibly, probably not with anything we have currently planned. That would be beyond HWO. I did show that we might be able to detect HDO versus H2O in using JWST spectra, but that really just kind of helps us determine whether water vapor has been lost from the planet, for example. But what you're talking about is whether we can do the sort of C12, O18 type stuff for CO2. I think that is more challenging. I think we looked at that for JWST and that was still extremely difficult. But back in the day, like 20 years ago, we had this concept of this thing called Life Finder, which was going to, potentially allow us to. In fact, they said they wanted to be able to image, waterfront real estate, and I said, never mind that. Just give me a high resolution spectrum. And so, if we get, better high resolution spectra, we might be able to do that. And actually, maybe the ELTs will be able to do that. It's something we should potentially look at is isotopic measurements with the ELTs. Yeah.
Okay, I might. Great talk. I might have misunderstood this, but the way that you're resolving the reflected light from the exoplanet and the light from its star is by making use of relative motion. You're looking at small dot ratios.
So there's a bunch of techniques we can use. One is sort of the brute force direct imaging, which is what HWO will do. It will actually spatially, angularly separate on the sky the sun from the planet. Right, which is fantastic. So we'll be able to. That's why it's this amazing new mission. So yes, we'll be able to. That's unambiguous. You'll be able to just get a spectrum of the dot that is orbiting around the star, right? Which is great. With the ELTs though, we can't resolve that. They do not have the capability to block out the starlight to be able to see something as faint as an Earth around it. So instead of doing that, they will. They will use coronography to help at least suppress some of the starlight. That's one step that's helpful. But then they will take the light and they will break it into a very high resolution spectrum. And yes, what will happen is that both the planet and the star and its planetary sister are moving relative to the Earth, and also the planet is going around the star. And so there are actual radial velocity shifts in the light that's coming to us from those motions, and we can separate that from the telluric lines that just sit where they are because they're in our rest frame, right? So the telluric lines from the Earth, the telluric lines sit there, and then you will see these things moving backwards and forwards that actually come from the planet and star system.
I think just for me and many people, I just would like to know, how does it affect the signal and noise?
Oh, it's really challenging, obviously. Yeah, it's incredibly challenging. But we're going to attempt it anyway. And some of the targets, like Proxima Centauri B, for example, nearby, might be doable.
They've done it for gas giants.
Yeah.
It's been very successful. So they have at least a baseline for understanding how to estimate light signal to noise for smaller, more challenging.
Yeah. I mean, this is incredibly difficult, and that's why I like to show that you can see that the onion skin of the Earth's atmosphere. That's what we're trying to probe. For a planet 10 parsecs away.
So let's give Vicky one more hand. Okay, I'm just bringing up your. I have to go. Share. Stop sharing. Sharing it down. Right. Did you get that right?
Okay, so that's hers. Yeah,
Good. Okay. And you are good with your mic?
I think so. Yeah, just
Is it turned on?
I think so.
Yes, it is. Okay. Great. Next presentation will be from Agouron from University of Washington. And you'll have a 10-minute talk. I'll give you an indication.
It's 8 minutes with 5.
With 2, yeah, exactly. So I'll give you an indication of 3 and then 1.
Okay, great, thank you. Yeah, can everybody hear me okay? Okay, yeah, thank you for the introduction. My name is Anna Grace. I'm a third-year PhD student at the University of Washington, and today I'm going to be discussing some of the research I've been doing over the last year, looking at exploring the detectability of Earth's surface biosphere from the Archean to modern day with applications to the search for life. Before I jump in, I want to acknowledge that this work came out of the Habitable Worlds Observatory Living Worlds Working Group, specifically the surface biosignatures on potentially habitable exoplanets. So one of the questions I think about a lot in my research is, are we alone? Now, this is obviously of astrobiological interest, but in the coming decades of observational astronomy, as we just heard from Vicky, we're going to inch ever closer to hopefully an answer for this question. And as of the beginning of this month, over 6,000 exoplanets have been discovered, 223 of which have been confirmed as terrestrial or Earth-like. So that means Earth-like in mass and radius. So now in this new age of exoplanet discovery and in anticipation of new discoveries to come, we ask ourselves two main questions. Number one, what planetary context is required to identify an inhabited planet from a lifeless one? And then as a natural follow-up to that, what technological specifications are actually needed to facilitate those necessary observations? Now, by technological specifications, I'm referring to telescopes, and one in particular, which we just heard a lot about, is the next NASA's flagship mission, the Habitable Worlds Observatory. So what I'm playing on the slide is a simulated solar system time-lapse observation taken at a distance of 33 light years away over the course of 10 years. So HabWorlds is being designed with astrobiology in mind, and in order to achieve its goals, hopefully we will be able to directly image around 25 terrestrial exoplanets in order to spectroscopically characterize their atmospheres and surfaces to search for signs of life. Now, by a sign of life, the technical term is a biosignature, which refers to any detectable feature of a planet that can be used as evidence for past or present life. And some of the well-known biosignature gases that Vicky was just talking about as well, carbon dioxide, methane, and molecular oxygen. So essentially for this project, we asked the question, what if Habitable Worlds observes a planet more like the Archean or even the Proterozoic Earth? Would we be able to tell if the surface is actually inhabited? So we know that for Earth's history, the biosphere has been mostly microbial up until fairly recently, but as life continued to evolve in the ocean and then on land, so did our reflected light spectrum, which is what I'm illustrating with the evolving figure on the left there. So in the following study, we conduct the first thorough exploration of the detectability of Earth's past and current biosphere, spanning from the Archean 4 billion years ago up to modern day. So to span such an immense amount of time, we chose to break it up into 7 distinct biospheres, which I'm sort of outlining in this table here on the left. And then based on geological proxy data taken for the biosphere at the time, we can choose a plausible surface biosignature to include in our retrievals. And then from this, we can forward model how the surface reflectivity, so reflectivity as a function of wavelength, has changed in reflected light over 4 billion years by parameterizing the planetary surface as a weighted mixture of both biological and abiotic surfaces. So following forward modeling is almost always a retrieval, and I'm including a corner plot here, the details of which aren't necessarily important, just to highlight that in addition to characterizing the surface of these exoplanets, we are also simultaneously retrieving on atmospheric constituents, planet mass and radius, and we also include clouds in our model. And this is for the vegetation red edge, so it's a modern Earth scenario. We started our exploration with the VRE partially or mostly because that's the dominant biological feature of modern Earth. So retrieval is essentially a way of fitting a model to data. So the inset figure there is where we have overplotted the true spectrum in blue with the retrieved spectrum in red, and we find that they match up well within uncertainty. And then from the retrieval, we can take a look at the extracted total forest fraction posterior that we find on the surface. This is for 15% of the surface covered in vegetation. And we find that in order to constrain that in an observation, you would need a signal to noise ratio minimum of 20 across a wavelength range of 0.2 to 1.8 microns. Now, generally a higher signal to noise ratio like 40, for example, is better in terms of reducing your noise, but it'll cost you a lot in future observing hours. So this is one of 5 retrieval grids completed for the study where we were testing what wavelength range was actually being needed in the future in order to achieve the goal of surface biosignature detection. And ultimately what we found is a minimum wavelength range of 0.4 to 1.1 microns is required. I'm going to break down the grid a little bit, starting with how we characterize this time period, as I mentioned before, with the 7 specific cases. And then for each case, we conducted retrievals at 3 different signal-to-noise ratios of 10, 20, and 40. And then within each signal-to-noise ratio, we also varied the amount of the surface of the planets covered in biology from 7.5% all the way up to 30%. So now zooming back out to the full grid, what we find is that a signal-to-noise ratio of 10 is insufficient to characterize the surface biosphere of Earth through time. For a higher signal-to-noise ratio, though, if you move to the right in the grid, up to around 20, things start to improve. And especially for the modern Earth case, which we call case 7 here, it becomes very detectable. From this grid and from the study, we've also found that the early and late Archean cases 1 and 2 are also comparable in terms of their detectability. So I've been focusing on the surface, but what I also want to highlight is that in all of the retrievals, we're also looking at the atmosphere, and some of these gases are biosignature gases. So this table is outlining which of those gases are simultaneously detected in our scenarios. And what we find is that all key biosignatures are simultaneously detected with the surface biosignatures, and this is particularly important for gases with several known abiotic formation pathways. Now, one of the final things we wanted to show is an agnostic method for characterizing the surface, where we want to fit the spectrum with a series of polynomials bounded by discontinuities, where the discontinuities can be considered an edge in the spectrum. So this work is highly preliminary, but I want to just briefly show that in our retrievals, we identified 2 edges, one in the near infrared and one in the visible, regardless of what parameterization we use. Now, the near infrared feature coincides with water absorption, and that's not a coincidence because there's also water in cells of vegetation. But the visible region feature is within the wavelength expected for biological pigment features. And I see I'm a little out of time, so I'll just walk through my conclusions here, and I'm happy to take any questions. Thank you very much.
Thanks for spending some time there. We have time for, I think, 1 or 2 questions here. Questions in the room or online, type your question. Do you have some? Anything online? No? Yes, Nikki. Thanks.
This is a really beautiful study, Anna Grace, and good job on all this wonderful work. So what happens when you have a more restricted wavelength range? Like if you can't get some of the shorter wavelengths due to ray rescattering, can you deconvolve the pigment from the abiotic surface?
Well, actually what we found is...
The question.
Yeah, the question was about what happens when you lose some of the ultraviolet information in the spectrum. And what we found is that surface biosignatures are largely insensitive to the short wavelength range. However, you do need to push more into the near infrared. So we did some tests looking at just the visible, and it was pretty catastrophic in terms of detectability. But we settled on this kind of nice range where you don't need to go too far into the UV and also not too far into the near infrared.
Okay, one more quick question if we have one, or we can save it to the discussion here. Yes.
Hi, can I ask why the modern plus lichen was so much less detectable than anything else, seemingly?
Yeah, so the spectrum we used for lichen, I mentioned this briefly, but our retrievals include weighted mixtures of biological and non-biological surfaces, and the spectrum of lichen was just a little bit too similar to like basalt and granite, so it was sort of getting confused in our retrievals, which I think also, from a practicality standpoint, on Earth, lichen's mostly present on those rocks as well, so it's just generally not a very detectable scenario, even with high SNR.
Okay, let's give Anna another hand. Next, we have a talk from Emily LaFlèche. Let me just bring up the presentation. Just put it over your ear.
Oh, like this one?
Yes, exactly. Right, I think we are good. We see that online, we're good? Right?
Can everyone hear me? Yes.
Yeah, so Emily's from Purdue University, and you, again, have 10 minutes.
Okay, sounds good. All right, thank you so much. Yeah, so today I'm going to be talking about my project, Detecting Seasonal Changes in Photosynthetic Biospheres on Earth-like Exoplanets. And this was actually submitted as a science case in the Living Worlds Working Group that Anna Grace mentioned. And I have some fantastic collaborators, many of which are here today, and some of which will also be here at AppSciCon next week. There we go. So Earth-like exoplanets, as was talked about in the two previous talks, are key targets for life detection with next-generation space telescopes like the Habitable Worlds Observatory. And what we're actually going to be able to do is observe, directly image Earth-like exoplanets around sun-like stars. So this is a really great step in the search for life. And we also talked a lot about how there are different types of biosignatures that we will be looking for, many of which are photosynthetic in origin. So for example, we have gaseous biosignatures like oxygen and its photochemical byproduct ozone, which is a really important one, particularly in the Proterozoic Earth scenario, because oxygen might not be detectable. And surface biosignatures like the vegetation red edge, which I think many of us here at the Extraterrestrial Photosynthesis Workshop are very familiar with. But both of these biosignatures can actually vary over time. And what we consider this is a different class of biosignatures called temporal biosignatures. And in particular, if these biosignatures are fluctuating on a time scale that we can actually detect with a telescope like HWO, this is really important for us to understand in terms of what the limitations are in detecting these features. So, like I mentioned, oxygen and VRE both vary seasonally. And in particular, this is because Earth has a planetary obliquity of around 23.5 degrees, which causes insolation and surface temperature to fluctuate in both hemispheres as the seasons change. This, of course, impacts photosynthesis, and it also changes the rates of photosynthesis with respect to aerobic respiration, which in the summertime can actually lead to a net outflux of oxygen from the biosphere to the atmosphere, as well as, obviously we have more leaves and grasses and other types of vegetative pigments that can be visible on the surface. But in the winter months, that changes. So we don't see as many leaves. The VRE signal is a lot lower. And we also see a net downflux of oxygen into the biosphere because respiration is higher than photosynthesis in these months. So this is a really interesting phenomenon. And one question that I'm really interested in is, could we potentially detect this seasonality on Earth-like exoplanets with HWO? So for this science case, we actually did two different case studies. The first of which is looking at oxygen and ozone seasonality, since they're kind of connected in that way. And we used first a photochemical model called PhotoChem, and then we calculated some gas abundances for both oxygen, ozone, and other gases in the atmosphere, and plugged those into RFAST to obtain some atmospheric retrievals and determine the detectability of this seasonal signature. So on worlds with low atmospheric PO2, very similar to the Proterozoic Earth, biogenic oxygen seasonality, so from the biosphere, can actually drive ozone seasonality. And this is particularly observable in the near UV. So you're seeing here the ozone absorption features in the near UV, and you're looking at essentially how in different resolving powers, we're looking at an R of 7 on the left and an R of 500, which is a little bit nicer and cleaner, but much higher than what HWO will be able to obtain, that we have different seasonal patterns in this particular absorption feature that might result in detectability of this seasonal signature. So the blue is like a winter scenario where you have around 5 ppm of oxygen, and that results in this particular shape of the ozone feature. And then in the spring and fall, you might have like 15 ppm of oxygen. It leads to more absorption. And then finally, in the summer, you have the highest absorption for around 25 ppm of oxygen in the atmosphere. So let's see if we can actually detect this with a Habitable Worlds Observatory-like mission. So there are two main things that we want to figure out in order to say whether this would be a detectable feature or not. First is, can we constrain the abundance of the gas in question, which is ozone? And that looks like essentially trying to get a posterior that kind of goes around this dotted line, this true value essentially that you can see there, where we would be able to say, okay, it's pretty likely that this feature is, ozone and that it has this particular abundance. But that's not enough to say whether seasonality is occurring. we also want to be able to differentiate these posteriors from one another from repeated observations of an Earth-like exoplanet over time. And so what we had to do here was both constrain and differentiate the seasonal ozone abundances in this near UV feature. And we wanted to see what SNR would be required to successfully differentiate between them. We saw, as you can see in the table down there with these green boxes, that for an SNR of around 20, we can actually pretty significantly differentiate these posteriors over the seasonal cycle. Obviously, it's a lot easier to do this between the summer and winter months where there's the most separation between those two posteriors, but we can potentially do this with adjacent seasons like winter and spring, for example. It becomes a little bit harder between spring and summer. But once we get higher than SNR of 20, it's significantly easier to do this. And so with these repeated observations to increase our signal with respect to noise, it's actually quite likely that we can do this. And this is a really interesting science case that HWO can explore. So our next case study was focused more on VRE seasonality. So again, now we're talking about surface biosignatures here, which are a little bit trickier. And so first, we used a model called CGD Plasm, which is a 3D Earth system model coupled to an atmospheric GCM that has a built-in vegetation module. And then we plugged in our vegetation cover data into RFAST to determine whether we can do the exact same thing I just explained, but now for the VRE. So here's our CGD Plasm results. And what we did for this particular case study is we looked at different obliquity values. So of course, like I mentioned, Earth has a 23.5 degree obliquity, which is pretty moderate. But imagine on an exoplanet that has even higher obliquity, it would have even more dramatic seasonality. And you can see that the vegetation cover follows suit from that. So if you have higher obliquity, you get greater spatial and seasonal variability in vegetation cover across the surface. And this will impact, especially our viewing geometry can impact this because it'll change the amount of vegetation that we can physically see in the illuminated portion of the planet over time. So this was really interesting to us. And we tested each of these different obliquity values for SNR 20 and SNR 40. And what we found is that it is indeed tricky to resolve this seasonal fluctuation in VRE, but it's not necessarily impossible. Particularly because the VRE is in the visible wavelength range, we're more likely to get better SNR in that wavelength range than something like the near UV for HWO. So we actually found that it might be possible to differentiate adjacent seasons, and really we're looking here at blue being spring and yellow being summer, and seeing those seasonal fluctuations of the VRE for an SNR of 40 or higher. So we would also particularly expect this on high obliquity worlds to be the easiest. So like an obliquity of 90, for example, which is our most dramatic seasonal case, we could potentially pull apart these seasonal fluctuations. So on my conclusion slide here, we've got that biogenic oxygen seasonality drives ozone seasonality, especially on low PO2 worlds like the Proterozoic Earth, which could be detectable for an SNR of 20 or higher in the near UV with HWO. VRE seasonality is trickier, but may be detectable particularly on high obliquity Earth-like exoplanets with an SNR of 40 or higher with the visible wavelength range HWO observations. However, in both cases, we need to emphasize that repeated observations of these worlds is necessary to identify these seasonal changes, and that it actually gives us higher signal and more contextualization of the biosphere, so we can actually really see the planet breathing, which is a really elegant way that Pin Chen described my project to me once, and it ended up being the title of my science case in the end, Breathing Worlds. So really what I want to emphasize as well is that these results are only a small subset of possible exoplanet cases. There's so much more that we can explore. And I'm actually working on some right now with different star types and how that could impact ozone seasonality, as well as different orbital configurations, viewing geometries, etc. So if you have any questions, please feel free to ask them now or email me. And then if you want to go ahead and check out our science case that we submitted, it got published recently, so you can scan that QR code and read more about it. Thank you so much.
Thanks so much. Time for a few questions. First one here.
Thank you. That was really interesting. So how much is the observed seasonality on Earth today? a product of our current climate and arrangement of continents. Like, would you see the seasonality signal during the Jurassic?
Right, so on like a Phanerozoic Earth, for my oxygen scenario in particular, we're not expecting to see a direct correlation between oxygen seasonality and ozone seasonality because there's a greater buildup of oxygen, which results in things like the ozone layer. So we're not going to necessarily see the same kind of detectable pattern. So like I mentioned, the cases that we're looking at are really these optimized cases for being able to look at this with HWO. But in terms of the VRA seasonality section, that is explicitly a modern Earth scenario, right? Because we're not expecting plant life on a Proterozoic Earth. And so it really does matter what seasonality you're referring to here and which wavelength range you want to look at to sort of be able to pull apart those features.
Sorry. So thanks very much for that. So I'll point out that Proterozoic oxygen is probably still 1 to 10% present at atmospheric levels.
Right.
So it's not trivial, and that raises the question of the baseline constraint. I'm sorry if you addressed this, but I'm thinking any planet that has enough biomass to give a detectable seasonal variation probably has enough oxygen in the atmosphere that this becomes an issue for signal to baseline.
Yeah, the baseline oxygen, actually we are testing that right now, so that's a great thing to bring up, that if you increase the baseline, it becomes harder to detect those seasonal variations in the ozone feature. But on an early Proterozoic world, we might consider that, you know, it would be a little bit lower. There are a range of Proterozoic oxygen estimates currently, like you mentioned, 1 to 10% PAL or present atmospheric levels is more late Proterozoic estimates. But early Proterozoic, we would consider possibly having lower than that. So we are testing multiple baseline scenarios to account for all the different features. Yeah.
Let's go with one more. We'll have to wait until discussion.
Hi, good talk. Can you comment on what type of how much obliquity we expect, and if we would be able to know how oblique a given exoplanet is?
Yeah, so in terms of how much we would expect, we don't really know, right? It kind of really depends on the fun little n-body simulation type of like orbital curve parameters that a lot of exoplanet scientists like to do. I am not one of those people. I don't mess with those types of things, but I know that in terms of detecting obliquity or being able to constrain obliquity for an exoplanet, that is an extremely large challenge. For HWO, it would be fantastic if we could potentially be able to do that. But the truth is we may not know what the obliquity of an exoplanet is for quite some time after its first observation. And so really what we're trying to do is analyze this for as many different obliquity cases as possible to be able to understand what those signatures would look like, quantify those kind of like posterior differences, and be able to link that back to the exoplanet. Even if we can't perfectly constrain the obliquity, that we would understand that some kind of seasonality is afoot. And there are other features that we could look at to confirm that seasonality is in fact occurring, both climatically and biospherically.
Thank you. Okay, let's give Emily another hand. So we have a shorter, slightly shorter than expected break, but we have 10 minutes now until 10:50 for coffee. And you can also, of course, find the speakers and ask them all your other additional questions. All right, see you at 10:50.